Enzyme kinetics, basically, as the term indicates, really is about studying or understanding how enzymes work, how fast they work, the rate of enzyme reactions, how fast an enzyme can convert a product to a substrate to a product. So, in essence, the most important thing to appreciate that first is the enzyme, the protein that makes up the enzyme, is the catalyst, which means that enzyme will catalyze the reaction of substrate to product, but that protein itself does not get changed. Okay? Very important thing. In order for an enzyme to work, it needs to first find its substrate. Enzyme, substrate. You form a complex, ES complex. Then the ES complex is going to go through a stage that is where the substrate is kind of activated. Okay? And after that, it's going to go through the product being formed, but the product will remain attached to the enzyme, and the last stage is the enzyme will release the product. So all of these sort of steps have their unique rate parameters or kinetic parameters. So K1, K minus 1, K2. You don't need to know much about them. I just bring them up here because when we talk about Michaelis-Menten kinetics, they kind of come in into a little bit of focus. So I just want you to appreciate that these rate constants will be eventually incorporated into a different constant. Okay, so what do enzymes do? Here's a reaction. Here's the energy scale, free energy scale on the y-axis, progress of reaction. So you start with A, you end with B. To go from A to B, you have to go through, let's start with the blue line, which is the reaction occurring on its own, uncatalyzed reaction. A goes through that transition state. You need what is called activation energy to make it to B. You take the same reaction, add the enzyme, note what happens. You still go from A to B, but you go through a lower transition state. The red double-headed arrow. The activation energy has been reduced. So that's exactly what enzymes do. They reduce the activation energy required from going from A to B. They don't change the energy of A or B, but just the activation energy required, and hence they change the rate of reactions. Okay? So you want to understand that. And like I said, there are steps for enzyme catalysis. The enzyme has to bind the substrate. Substrate goes through transition state. Complex of the enzyme starts to work on that substrate, and it eventually makes the product, but the product stays attached to the enzyme, and eventually the product is released from the enzyme. So there are multiple steps involved, and we'll go through them here. Pretty straightforward. So here's your enzyme. You're going to have this big sort of cleft here, or domain if you want, this large one is called the binding site. Much larger, maybe 100, 200 amino acids. And within it, there's going to be a small area of maybe 10, 20 amino acids are the catalytic sites. That's where the substrate is changed. However, the association of the substrate with the enzyme requires a larger area on the enzyme, the binding site. Okay, so binding site large, catalytic site smaller number of amino acid residues. So here's the substrate, binds, you form the complex. As you can see, you form now the activated transition state where you have ES star. That means the substrate is beginning to be changed. You can see some changes there. It's neither substrate nor quite product yet. So that's the transition state. Then it is converted to product. Two new molecules have formed, but they are still attached to the enzyme. And the last step is to release the product. In this case, the product are two new molecules. Okay? So as I mentioned, you need really two important parts of the enzyme: the binding site and the catalytic site. The binding site is a larger, if you want, pocket or cleft where the substrate is situated in. Typically, that's an example of a domain. Typically, that may require about 100 amino acids. But remember, this is part of the tertiary structure. So these amino acids may be from different parts of the protein after it has folded. They line the pocket where the substrate is going to fit in. Importantly, the substrate and the enzyme will interact through non-covalent interactions of the substrate with the R groups that are lining up that binding site. Okay? No covalent bonds, just interactions between the substrate and the R groups that are exposed in the binding site. Within the binding site, there may be a very small number of amino acid residues that are involved in catalysis. That means they are actually going to be part of changing the substrate to product. So this is much smaller site. This is more in terms of situating the substrate in the pocket in the right way so that the enzyme can work on it. So I just covered that, and really most of the interaction, all of the interaction is going to be non-covalent between the R groups of the binding site and the substrate. The old model was a lock and key model. We don't really accept that. It's no longer accepted. So the new model is more of a sort of dynamic induced fit model, meaning here's the enzyme and here's the specialized pocket. It's more dynamic rather than rigid. It means it can sort of change its shape and accommodate the substrate so the fit to fit it that way. So that's the more likely that the binding site has a dynamic characteristic. It can change its conformation to accommodate the substrate. So the things that affect enzyme activity are many, including things we already talked about. First of all, we talked about metal ions, cofactors, and some enzymes require those to work, like magnesium, manganese, copper, zinc, and so on. Some enzymes require effectors, allosteric effectors, that can change the activity. They bind to a site besides the binding site, the substrate binding site, and alter how the enzyme work. So we've covered that a little bit before. And here are the environmental factors, meaning things like temperature, pH, things like that. Remember, enzymes are protein. And proteins are in their native structure usually within a certain temperature range, within a certain pH range. Okay? So I'm gonna focus first on pH. So pepsin is an enzyme found in the stomach. The pH of the stomach is about 1.5. So you find that this protein, this enzyme protein, is quite resilient, and it works in the acidic environment of the stomach. On the other hand, trypsin, which is derived from trypsinogen, is in the small intestine. And look, it works roughly around neutral pH. This is the enzyme activity, and this is pH. So that the higher this is, that's the enzyme activity, velocity here. So you see trypsin is working in the ambient environment where it is produced, whereas pepsin works in the ambient environment where it is found. So it usually makes sense. If you take trypsin, put it in the stomach, it won't work. If you take pepsin, put it in the small intestine, it won't work either. So that's the importance of pH. So the ambient environment, the ambient pH is quite important. And you can see there's a spectrum of pHs where different enzymes work on. Again, don't memorize anything. Just appreciate the importance of pH in the ambient environment. Okay. Temperature, again, if you look at most enzymes, at least mammalian enzymes, they work in this range, around 25 to 40 degrees centigrade. Below it and above it, especially above it, below it they get sluggish, you decrease enzyme activity. Above it, you start to denature the enzyme. Remember, enzymes are protein. You denature them, they no longer function, and they become irreversibly inactivated.